Bruce said, this is kind of an emotional talk to give. I think I qualify for that as well. So here's my title, and that title or my profession or something has prompted a couple of questions. So the first one is, where did I go wrong? It's been asked of me several times today. And I think I can trace the beginning just down the hill and also down the road from Martin Saunders. So we grew up a couple blocks from Martin, and Rudy Zallinger lived on the same street with him and did the wonderful murals in Peabody Museum. Between the murals and the skeletons, I was absolutely fascinated with dinosaurs. And so I sort of made the progression, started out interested in herpetology, and then got more generally into biology. So I can blame that for where I started to go wrong. I think it was pretty clear I had gone wrong by graduate school. My PhD thesis was on an enzyme alanine racemase. So to me, there's sort of nothing more antithetical to the son of a synthetic organic chemist than saying, I'm going to destroy all chirality as a business. So, but I think my father would have put the moment somewhere in between. I was an undergraduate at Harvard and had the pleasure of having Frank Westheimer take the occasion to teach the entire year of organic chemistry. And at that point, I still wasn't sure what I wanted to do. Having two parents who were organic chemists, I was quite excited. I got into the course. And on one occasion, I called home to tell my father about what I had learned in class that day. And it's actually a reaction that's been described by two of our speakers today more accurately than I described it. So I called up and I said, yeah, I learned about the Niels-Alder reaction. So that kind of blew my cover, and so. Anyhow, though, I'm willing to sort of help people out here. So let's call this a natural product experiment. All right. So the second question is, Steve, will I understand anything you say? And so I'm gonna try hard. And you know, one thing that should have been clear from the talks, everything's been in black and white so far, and I didn't realize why. This is what the MIT chemistry lab was like. So you guys all trained in black and white as far as I can tell. And this is actually this is the undergraduate lab at MIT where Harry must have studied. But I want to reassure you, okay? First off, my father did appreciate the interface between chemistry and biology. So this was on the back of a letter he sent me, a catalyst. And I unfortunately, my daughter's in the audience, and she knows all too well that the tradition of punning has sort of been handed down from Harry to children to grandchildren and such. So that's the way things are. But scientific method, okay? So we're biologists. We do things in fairly similar fashion, all right? We make observations. We have hypotheses. We do experiments. We conclusion such. So this is not just chemistry, but it's biology as well, okay? It just looks a little different when we do it, okay? So this is my lab research group at one point. And so we've been meeting. And this is a write-up of our proposal. We've made our predictions. So Matt here was responsible for holding that. And here's the experiment in progress. So people ask, why didn't you end up at Yale? I'm at University of California, San Diego. This is very convenient for behavioral biology. Okay. So this is what we spend more of our time working with. It's the fruit fly Drosophila. And we study it because unlike thoroughbreds, people, various things, we can do genetics. And that's really the core of the way we go about things, is to use the process of genetics to make sure we're studying what we think we're studying in. And often those things are chemical, or at least biochemical, I would argue. All right. So I'm going to talk about two different questions which any of us, at the time we started working on them, would have been thought to be completely separate. And they're basic biological questions. So one is, how does a single cell that's just kind of a football shape turn into something like all the people sitting in this room, arms, organs, nervous system? How do those instructions, and we ask it at a very basic level. We start out with just, how do you get a front and a back? Okay. So this is a fruit fly larva. It needs to eat. It crawls along the food, and so on its belly, it has these little triangular-shaped denticles that it can use to crawl on the food, and its back, it doesn't need those. So ventral, the belly, dorsal, back, they're different. And mutations were first found that affected this process in the lab of Christian Nusslein-Volhard, who did some other genetic work on the anterior-posterior axis that won her and Eric Wieschaus Nobel Prize about a decade ago. So other individuals following up on her work were able to map out some of the molecular components and show that the way you get ventral different from dorsal is you make one gene turn on only on the ventral side. So here the blue gene is being expressed, the red gene is being turned on only dorsally, and there's a gene that's at the interface there. And what specifies that is this gene regulator transcription factor called dorsal, and it goes into nuclei on the ventral side. So this seems completely backwards. It is, because it's genetic. So the first mutant back a century ago was called white. It was because fruit flies with that mutation had white eyes, and the white gene is responsible to make eyes red. Okay? So when the white gene's gone, you get white eyes. So when the dorsal gene is gone, you only get dorsal tissue, and that's why the name is completely backwards. So we study then, how do you get this gradient? These are all nuclei. It's one cell with several thousand nuclei, but the transcription factor is only in these nuclei on the ventral side, turning on this blue gene. It's not a nuclei up here, so the red gene can be turned on by its absence. Okay. So that was one line of research. Meanwhile, there was a gentleman named Hans Bohmann working in Sweden asking a very different question. And that question is, how does an organism identify and eliminate non-self cells? Okay? So when you study biology, you learn every cell has a plasma membrane, and it's got a nucleus, unless it's a bacterium, and we all have the same genetic code. So how do you figure out when something's not your own cell? Okay? And so that's sort of the fundamental question in immunity. And what Bohmann did was start studying this in another insect. This is, any of you have ever seen a Cecropia moth? It's about this big, they're beautiful. This is the larva, which is also gorgeous. And you may ask yourself, so why did he study it in Cecropia rather than in something, a more standard thing like the Drosophila? And the answer is, that's a Drosophila next to a dime. I know many of you in the audience have an iPhone and never seen a dime, but that was a form of currency, and it's actually a lot smaller than that. That's a fruit fly that's a lot smaller than the dime, and an egg would be about the size of the pointer spot. So that's the size of a larva. This is the size of a Cecropia larva, okay? So they keep, in the old days, Havana cigars used to come in these big, long boxes or cans, and they grow these. They actually give one larva per box or can. So you can do biochemistry. You can get enough material. And so what he did was to grind these up and then purify the proteins. And I'm going to show you sort of a nice biological twist on a procedure that I think many of you will know. That's to do gel electrophoresis and to stain the proteins that are being separated. So these are the purified antimicrobial peptides, but they're not actually being stained here, even though it looks like it. What Don Holtmark in Beaumont's lab came up with was to separate all these materials in this electric field, in this acrylamide matrix, And then after he was done, instead of applying a biochemical stain, he just spread bacteria over the entire gel, put it in an incubator overnight, and then shone light on it. And so what you're actually seeing here are the places where there's no bacterial growth. So it was a very nice assay that could monitor purification, and it led to the discovery of this system of defense called innate immunity that all organisms have. So in human, anybody here who took human biology more than about 15 years ago almost didn't hear about this. It was this primitive immunity. It was the invertebrate immunity. But in fact, it's turned out to be fundamental in all organisms. Our lymphocytes, our B cells and T cells and antibodies don't get made unless this system provides the information. Plus, you got to wait about 2 weeks for that system to kick in, and this is what keeps you alive until then. And so the Nobel Prize 2 years ago now was for discoveries in this basic innate immune system. So these seem to be completely separate areas of research, but in fact they're exactly the same, and they're the same in rough terms all the way from fruit flies, shown here, to humans over here. So those are the two species in my title. And these are the three sort of basic components. First is you have a receptor. You have something on the cell surface that can receive information and say, this is non-self. So bacteria have cell walls. If you detect a piece of cell wall, that's non-self. Viruses have double-stranded ribonucleic acid, RNA. If you detect that, it's a virus. It's non-self. So these systems detect molecules that microorganisms, fungi, bacteria, viruses have that we don't. And they relay that information into the cell. There's a series of biochemical steps, and they lead to the release of this transcription factor. You heard about one called dorsal in the embryo. This is called dorsal-related immune factor. In humans, it's called NF-kappa B. And what keeps that transcription factor normally from turning on genes is an inhibitor protein, cactus in Drosophila or bikappa B. So the basic outline is completely identical in all the way from flies to humans. If you look in detail, there's more similarity, but there are also some differences. So there are adapter proteins and various things. And I'm going to talk about our work on some of those differences and how they've revealed sometimes a more greater commonality than we had expected, or a difference that was unanticipated. So I'm going to start with these genes, tube and pella, that we were working on. And we used molecular tools to identify the genes that were affected in the mutations. And when we looked at the proteins, we found that they both had a death domain, okay? This is kind of like a Death Star. Not really. What happened is somebody was studying a pathway that is involved in signaling cells to die. So we're all, when we're embryos, we all have a little tail. That you got to get rid of that because we don't need a tail, unlike frogs or unlike tadpoles, right? So we get rid of it earlier. And there's a pathway that says die cells, and that's called the cell death pathway. And this kind of region was first found there, and we found it in both this protein pella and this protein tube, and we found that these two could interact. And nobody had seen a structure for one of these. We collaborated with our colleague then in Dallas at the medical center to get such a structure, and this is what it looked like. So you can see sort of three pairs of alpha helices, and this is a head-to-tail configuration. So normally we used to always sit around and go like this and think about dimerization, but actually this is this way, the fingers, and that's going to be important in a moment. Okay. This is a beautiful structure, but we had a big problem, and this is where genetics came to play. So the big problem we had was, does this have anything to do with what's going on in biology? There are lots of proteins with death domains. Sometimes you stick proteins together that are similar, they will stick. Did these proteins actually interact this way in the embryo or in the immune system. And the evidence was almost contradictory because when we used antibodies, the two proteins were very rarely in the same place. We couldn't use one to drag down the other in a biochemical experiment. So we really needed to go out and prove that this was the case. And here's where sort of, I think, being brought up by chemists made a difference, as you'll see in a moment. So we went to the crystallographers and we said, what's important here? What's the key structure? And they said, well, there's a lot of binding energy. See, this is a term that you recognize. A lot of binding energy here. This is negatively charged, this glutamate. This is positively charged, this arginine. So there's an ionic bond, and we think that's important. So we said, okay, if that's the case, if we make a mutation and we convert that glutamate to a lysine, it'll now be positively charged. We should get charge repulsion. We took RNA that encoded that mutant, put it into that mutation, put it into mutant embryos, and it didn't work. We said, well, let's be sure. Let's try the other one. So we changed that arginine to a glutamate. This, again, should not work, and that was it. So that's great. And that's normally where genetics stops, is to sort of give you supportive evidence and presumptive evidence. But the beauty of having a crystal structure is we could go one step further. We could say, well, what if we change them both? Okay? Both those proteins are dead, but we've effectively just flipped the dipole if we put both. So we injected both dead forms, and they compensated. Okay? And I'll tell you actually sort of a side story at Yale that we were inspired in this experiment by an experiment done by Charlie Yanofsky, who was started his career here at Yale, and who my mother, Elga Wasserman, actually worked as a research associate shortly after Harry came to Yale. And Yanofsky did this with compensatory changes in nucleic acid. Here we are doing it with two different proteins. And I'll show you biochemical evidence for this in just a moment. Okay. So when you combine the genetics and the biochemistry, it's now much more powerful than either alone. The biochemistry said these things can associate in this way. Genetics says this site's important. When you get the compensatory change, it says these two proteins interact in exactly this way in vivo functionally to make this happen. So that's the important structure. Here's yet another death domain protein, this protein over here, MYD88. So now what happens? Are they alternate pairs? Are they a triangle? Are they a stack? How do these three proteins interact? So now we could turn to cultured cells. So these are cells in tissue culture. And we could put all three of these genes in that cover all three of these proteins and ask what happens. So here, if you put in all three genes and use an antibody to one of them, all three are associated. So they're all bound together. If you use either of those mutations I told you about, MYD88 you're bringing down with an antibody, tube comes along for the ride just fine, but now Pella is not coming down. And yet those mutations affected where tube and Pella interact, one of them is in fact a mutation in the tube gene. And if you put the compensatory pair together, now you restore association. And so the way we interpret that is that Pella doesn't bind to MYD88. It can only bind through its association with tube. And so the mutations here are affecting Pella because they're affecting Pella's binding to tube. So we have this trimeric structure, and now we wanted to ask, can we analyze it in more detail? And what I'm going to show you is just take you through a little of the biochemical nitty-gritty, because where it's led us is to be able to ask questions about evolution, something we really never anticipated when we started this study. So MYD88 must bind on the other side. We made a series of mutations all along the gene, and three of them, quite far apart in the primary sequence, all affect the ability to bind to tube. When you look on the 3D structure, they're not far apart at all, and they're on the opposite side from the mutation that affects binding to Pella. So now we've got all these interactions mapped out, and we have a real puzzle on our hands, because what I've shown you is that tube has very specific interactions with this protein, very specific interactions with this protein. You look over here, there's no tube. And so what's going on? Okay, where did tube come from? There are two sort of fundamental possibilities if you think about evolution. One is tube was there up in the ancestor, and it got lost, but how did you get rid of all those very specific interactions and still have function? The other is, it wasn't there in the ancestor. In that case, how did you stick this in? Okay? So sort of a fundamental evolutionary question. And what was delightful for us was that with the Human Genome Project and other genome projects, we started to have all kinds of data available. And so we asked, what about other insects and other invertebrates? And we went looking with the death domains of tube and pella, and we found lots of things. So we found things, so tube has these repeated regions, pella has a kinase domain, and we found things with repeats and a kinase domain, with a kinase domain, with nothing. And we sort of scratched our heads, how can we make sense of this? And this is where all the mapping we had done of interactions became useful, because we said, well, we know that if you're a tube protein, you bind MYD88 over here, and you bind pella over here. So we said, let's take those criteria and look at the genes we've discovered. And what we found is that some of them had conservation of the regions that bound MYD88 and conservation of the regions that bound pella, and others did not. So then if we classified things and colorized them, suddenly we had Noah's Ark. Okay? Everybody has a tube, everybody has a pella, everybody gets a car. Okay? So, and this was true in mosquitoes, in honeybees, even in mollusks. And that was provocative, because what we realized was that there were these genes that had been described in humans called IRAC, for their kinase associated with a particular receptor, and everybody, ourselves included, had always thought they were just forms of pella because they didn't have these repeats and they had a kinase domain. But it turns out that of this green family, only in Drosophila melanogaster do you find the repeats and nothing else. All these others have a kinase domain or kinase domain and repeats. So it was an accident of the organism we had started with that we didn't see this possible connection. So we then analyzed these two proteins. It turns out IRAC1 looks just like pella, and IRAC4, by the criteria we had, looks like a tube. And so now we went back and said, okay, here's the right picture. There was, in fact, a very fundamental conservation, and nobody had appreciated that there were two classes of IRACs, and the architecture is, in fact, the same all the way from flies to humans. And this was confirmed when there was a crystal structure done a few years ago, and they found MYD88, IRAC 2 and 4, and that those coordinates could be mapped right onto the structures that we had found. So the basic architecture is all the same. So that's a story where things looked fundamentally different, and through first a molecular genetic approach and then an evolutionary approach, we could show there was in fact conservation. So let me turn now to this question. Why do you have two of these, and where did they come from? So all these organisms seem to have two death domain proteins, many times with a kinase. The simplest model would be that they arose by divergence. Okay? That there was a protein that had this interaction domain, a kinase domain, and then you made a copy of it. And one copy became one class, the pink, purple, mauve class, and the other the green class. One the pellas and one the tubes. And what was delightful was we could now test that. We could say, can we run evolution in reverse? Okay? And to do that, what we did was take the gene for tube and the gene for pella and fuse them together. We made something that had the repeats, but also a kinase domain. And then we put that into organisms that had neither tube or pella. In other words, we had a double mutant in Drosophila, had no tuber, no Pella function, and lo and behold, this is what we saw. So if you have neither gene, you're just a sac of back, okay? You're dorsalized, there's no ventral tissue, there's none of those denticles that we saw. But if you put in this thing that's a combination of tube and Pella, now you get nearly complete pattern. They don't live, but they're nearly wild. And so we could therefore demonstrate that a gene with the ancestral gene, in fact, what we think would be the ancestral gene, that almost all that function is still there. Okay. So why duplicate? And here's a question for you chemists. So it turns out that there's a catalytic residue and aspartic acid, and classically there's an arginine in front of that. So most kinases of this type are called RD kinases. And these Pella kinases instead have a glycine, and that's only seen in one other place in any numbers, and that's in plant kinases that are also involved in defense. And what we think is that they're involved in kinases not that catalyze cascades of phosphorylation, but rather ones that promote assembly of structures. Okay. So, I'm going to tell you a whole new story, so if you're lost, you get a chance to catch up. A whole new story in this pathway, and that is focusing on this other difference here, and that is there's this complex of kinases, associated factors, and such that I'm not showing on the fly side. The fly, in fact, has them. If you look at the genome, they're there, but they have nothing to do with this pathway. You knock them out genetically, this pathway works fine. And we were quite curious about that. This system here is, in fact, an active target in many biotech companies for anti-inflammatory drugs. Many of the diseases, arthritis and Crohn's disease, colitis, a lot of inflammatory disorders, in fact, arise from overactive components of the innate immune system. Basically, you're fighting an infection that's not there and end up attacking your own tissue. So this is quite a bit of interest. The question is how the fly is operating this pathway without it. What was really surprising is, so here's the gene IKB on the human side, cactus on the fly side. They look very, very similar. Okay? These are sort of protein motif structures called anchor repeats. Here's this sequence with two critical serines. Here's a sequence with two critical serines. Here's a protein degradation signal. So these things, for all intents and purposes, look like they should work the same way and be regulated the same way. And if we, here's the wild-type cactus. When you turn on the pathway, this inhibitor gets degraded. That's what allows the transcription factor to go into the nucleus. If you mutate these serines, it doesn't get degraded. So that's, again, similar to what had been seen on the human side. So we were really quite puzzled as, and there's some additional serine that can be involved. We were quite puzzled as to why these things that are so similar are regulated in such a different way. And so everybody assumed, well, we just must be missing a component of the pathway. And three different groups, one in Boston, one in Germany, and one in Sweden, went looking genetically comprehensive. Nowadays, you can go in and mutate every gene in the organism and ask what happens. And they couldn't find another kinase. The only thing they found was the gene Pella I've already told you about. This one operates downstream. So in humans, you have this gene, IRAC. It's not enough. It needs this whole complex. In flies, you have this gene Pella, and it appears to be enough. How does it work? And the problem is that when we started trying to make this kinase, make a biochemical preparation, it caused lethality in bacteria. And I think, again, here is a place where my organic chemistry roots came into play. So we had a problem that we're making this kinase, and if you make it enough bacteria, it must put phosphate groups on some proteins of bacteria that don't like it, and it kills the bacteria. So what we did was we couldn't put a protecting group on every protein in bacteria, but we decided let's get rid of the unhappy byproduct. So we took advantage of bacterial biology where often genes are made in tandem, and we cloned the pella gene and then a phosphatase right behind it. So basically we've got a bacterium full of a kinase. Bacteria normally don't have those. It's doing all kinds of nasty phosphorylation, and now the phosphatase is running along behind it, taking all those phosphates off. And the phosphatase is completely acting at random. It doesn't care where a phosphate got put on. And sure enough, that allows us to express this in high quantities without any deleterious effect. So we purified the pella, and we asked, can it in fact directly phosphorylate this protein cactus? And the answer is yes. This is a radio-labeling study. You see phosphorylation. And then if we mutate just 3 of the 50 serine residues here, which are exactly the 3 that we knew were required for signaling, we just about wipe out the activity. That says it's necessary that pella can't act on this unless those serines are there. But could we in fact show that it phosphorylates these serines directly? And here we could take advantage. So here you see it phosphorylates a mutant where those serines are gone, pella doesn't phosphorylate. A mutant where the pest serines are gone, gets phosphorylated by pella, doesn't get phosphorylated by a separate kinase. So reciprocal activities. But here we could put the N-terminus from the human protein onto the Drosophila protein. And the beauty of the human protein, because so many people are interested, they have all kinds of great reagents, including an antibody that's specific for the phosphorylated form of these 2 serines. And lo and behold, when we make this chimeric form, our protein from Drosophila is phosphorylating those residues just fine. So I will finish up the science here with on this question with another sort of biochemical poser, which is what I've told you is that the sites are conserved. That's why everybody assumed the kinase would be the same, but the kinase is completely different. So why would you have the same signal in the cell that says phosphorylate here when you use a different kinase? And you can see it's the only thing that's conserved. So here are the serines. Those are here's 1, 2, 3. Basically, nothing else is the same. And here, when you look in flies and mosquitoes and flower beetles, this region is completely the same. So why is it the same? And the answer is we're looking at it wrong. Okay? Yes, it's a site of phosphorylation, but that's not why it's conserved. It's conserved because the degradation machinery recognizes those serines when they're phosphorylated and only recognizes them in this context. So here, from different pathways in different organisms, the pella kinase, I-kappa B kinase, the 2 I've told you about, but also GSK3, CK2, and these you can see targets in a variety of different proteins. So what we were fooled at by is seeing this conserved sequence that gets phosphorylated. It's not conserved, and because it needs to be recognized by the kinase, it's conserved because after the kinase acts, once it's phosphorylated, it has to be recognized by this machinery. And we could show that genetically, where if we knock out one component of that, we stabilize the protein. So let me just leave this dissection of the pathway here and talk for 5 minutes about what goes on downstream, which is where we are nowadays. We sort of feel we've mapped this out pretty well. And so fruit flies, many of you I think have encountered fruit flies, and they in fact live in lots of different places and very different environments, and where you can imagine they would encounter very different pathogens. So I never knew there was a name for this. I mean, I think we've all walked around. I was just up in East Rock Park today, where you see trees with split trunks and this stuff. Anyhow, that's slime flies, right? And that's a bacterial slime fly, so it's a really good name for it. Yeasty fruit, that's what you That's why they're in your house, because they're waiting for the fruit to rot. These guys in the tropics just have a really good life. And these have the tough life. The mojavenses, they live out in the Mojave Desert, and they live on a cactus rock that nothing else can survive, but they've evolved to be able to tolerate. So we thought, all right, if we want to study how an immune system works, then we ought to be able to compare the immune systems of these 4 different Drosophila species that live in very different places. And they're quite divergent in terms of evolutionary time, so that should allow us to get some good information. And so we don't want to actually sit there and stab them with pathogens, so we use this biopesticide called Bovaria bassiana, and it's a white powder spores, and you can see it takes out spiders and grasshoppers and flies and beetles. And so I called up this guy in Arizona who sells the stuff. I said, can you send me some? He said, sure, but it's really potent. I thought, we've got all our fruit flies, all our genetic mutants. I don't want to have them wiped out, particularly because a lot of them have purposefully damaged immune systems. So I said, send it to my buddy Randy. And so what I effectively did is, and so he sent this, but he didn't send it officially. He basically sent a white box, inside of which was a vial of white powder, okay, to an address that he didn't know, and therefore the person receiving it didn't know. And fortunately, I somehow mentioned this to my buddy Randy in a conversation along the way, and fortunately as well that the FBI did not come crashing down his door at the point that he was ordering white spores in the mail. But this worked for us great. We did what's called RNA-seq, and I'll give you the punchline here. I mean, that's not a type of 7 billion base pairs of information from one experiment. And so now in a biology lab, you basically have to have somebody who handles data. So I have a great postdoc who use these programs. Also in biology, we're all cutesy, okay, right? I mean, so the guy who wrote the software, it's bow tie, top hat, cufflinks, cuff compare, okay, so everything. And he's at Berkeley, so. And it's incredibly powerful. It tells you where genes are and where they're not, and when they're, here we can look at uninfected versus infected flies. And I'll give you just the punchline. So here, this is a Venn diagram. So overlaps are indicated with different colors and such. So here are the 4 species. And what this tells you is that there are 47 genes we found only in Drosophila melanogaster. And at the other extreme, there are 17 genes that were in all 4 species. And what was amazing to us was that all the genes that we know about how they work in innate immunity were not in this 17. Those 17 people hadn't studied, hadn't recognized as important, and yet this tells us they're really the core thing. And so we've used a new technology which is called TALEN. There are these 2 protein approaches called TALENs and CRISPRs. You can basically go in and design away genes, move things around and such. And what we found is they in fact have genetically an essential role in the toll response. So what we're doing now is dissecting out that role and trying to find exactly their biochemical activities. And by we, what I want to say is, here's the crew. So this is Scott, who's the really talented bioinformatician, Alexa, a grad student who did the work on these effectors. She's just about to graduate. And the work on the signaling pathway upstream was a visiting grad student from Norway, Liv, and a technician at that time, Jesse. So I'll stop there and make it a little less rushed for the next part of the presentation. So thank you.